Hello, good morning. How are you? Fine. Let us continue our voyage into the experimental pursuit of Gregor Mendel, father of genetics. In the previous class, we have seen how genetics is evolved, what is genetics, what is heredity, what is variation, what was the experiment conducted by Mendel in Pisum sativum, and what made Mendel to select this plant as his experimental tool, and how he could derive his results after 8 years of continuous research on thousands of plants. And what was the result? What was the fate of his findings presented before the Natural Society of Brunn, and how he was actually recognized after 18 years of his demise in 1900 by 3 different scientists at different corners of the world: Hugo de Vries, Carl Correns and Sherman, and how he was actually given the title Father of Genetics. And today let us see some key concepts, important concepts in genetics. First one is called allele or allelomorph. Okay, we get to spell in alleles or allelomorphs. So there are actually the factors representing 2 alternative forms of a trait. So they are the alternative forms as the 2 sides of the leaf, or 2 sides of the same coin. Okay, they are called alleles or allelomorphs. So, for example, if we are considering the character height of the plant, it can be either tall or it can be dwarf. So, height of the plant is the trait. Height of the plant, and it can be either tall or it can be dwarf. Okay, it can be either tall or dwarf. So they are called alleles, and these alleles are normally represented by English letters. If it is a tall plant, it can be capital T, or it can be capital T and small t. And dwarf is always small t. Okay. So these are the allelic forms. So the alleles are the alternative forms of the very same trait. Alternate forms of very same trait. So here we have the capital T. It can be, the condition is called the homozygous condition. If both the alleles are of the very same type, it is called homozygous condition or state. So we have the capital T as homozygous condition, as well as small t again. That is also homozygous condition. But here the 2 alleles are of different types. One is capital T and the other is small t. It is called the heterozygous condition. Heterozygous condition. Okay. So the 2 alleles are present within the organism, individual. It can be either in the homozygous condition or it can be in the heterozygous condition. Then second concept is gene locus. So it is a particular gene of a chromosome representing a single gene. Okay. You know what is a gene? You have studied in your plus 2 level. What is a gene? So gene is a functional unit that will produce a character or a trait. So there are several genes present in the entire chromosome. Okay? There are several genes present in the DNA. Okay. And we know, because we have in human beings we have 23 pairs of chromosomes. 23 pairs of chromosomes. But the number of traits or characters exhibited by human beings, they are of several times than this 23 pairs. Okay. People sitting in the class, you people sitting in the class, each and everyone will be having different temperaments, different abilities, different characters. All these belongs to the DNA or the makeup, genetic makeup of you people. So, the gene locus is a particular region of chromosome representing a single gene. So, the alleles of a gene occupy the same gene locus on the two homologous chromosomes. Okay? So, we know normally there are two chromosomes or two sets of chromosomes in each of us. One set, 23 chromosomes coming from the paternal side, and the other 23 chromosomes coming from the maternal side. These two, 23 chromosomes, they are pairing with each other and they are called homologous chromosomes. Okay, homologous chromosomes. And both the chromosomes, homologous chromosomes, they will be having one allele each for a particular trait. So you will be now understanding how it is. I have written this capital T, small t, capital T, small t. So here, one allele is coming from the maternal side and the other is coming from the paternal side. Clear? Here also, one will be coming from the paternal side, the other from the maternal side. Okay. So in any character trait, one allele will be on one homologous chromosome coming from the father and the other will be from the mother. So these two alleles are coming from either side, father and mother. So in a character trait, there will be two alleles, and one will be from the father and the other will be from the mother. So that is the gene locus. Then what are factors of genes? So factors are called by Mendel. Okay. So Mendel called these factors which control the expression and transmission of characters. So these factors were later termed as genes by Johansen. So at Mendel's time, molecular biology or even microscopes of good quality was not at all there. So he called some factors. So he could make out that some factors are present in the organism and these factors are responsible for the expression and transmission of characters. He did not know what is chromosome, what is gene, what is DNA, whatever. So he called it as factors and these factors are responsible for characters. That was Mendel's conclusion. And Johansen later on termed these genes as factors. Okay, these are very important concepts in genetics: alleles, gene locus, and factors of genes. I think it is clear. Then another supplemental concept is dominant allele and recessive allele. Okay. So again coming to what I have written here, you may be wondering what this capital T, small t, capital T, small t. Okay, so capital T and small t. So these are the two alleles responsible for the character height of the plant. As I said earlier, height of the plant can be either tall or dwarf. And one of the pairs of alleles, that is capital T and small t, one of the alleles will be expressed in the hybrid in the next generation, and the other will be suppressed or masked. It is not expressed. So the expressed allele is termed as dominant allele. The allele or the allele expression which we have seen, we can see in the next generation, that allele is called dominant allele. And the one which is not expressed in the immediate progeny, and that allele is called recessive allele. A recessive allele doesn't mean that it is dead or it is deleted. It is there, it is present there, but it is not expressed. It is dominated by the dominant allele. You may have noticed the experience of the cock, male member of a poultry, cold-cooings. If there are several male birds, only one will be cooing. All others will be keeping mum. Have you noticed it? Yes. And once this dominant male animal cock is being killed, immediately the next one will start cooing. Am I right? Yes. In every form of animals, there will be one dominating member who will be ruling all of this. Take the example of a class. It may not be so appropriate. There will be a leader, unelected. He or she may act as an ordinary leader. She or he will be dominating the other class, and all others will be agreeing with all. Okay. So in genetics, the allelic form which is expressing in the next generation is called a dominant allele, which is represented by this capital letter, and the one which is suppressed or which is unable to express in the next generation, the next hybrid, it is called recessive allele. A recessive allele doesn't mean that it is dead, it is gone. No. For the time being, it is being keeping silent. And if there are only two recessive alleles coming into a particular hybrid, naturally this recessive character will be expressed. That means in recessive condition, homozygous recessive condition, the character of this recessive allele, that is dwarfness, will be expressed. And if a character is expressed, it doesn't mean that it is dominant. So a recessive character also can express in the next generation, provided they are in the homozygous recessive condition. And a hybrid, in simple terms, a hybrid is an organism produced after cross-pollinating two genetically different parents or individuals. So genetically different two individuals are closed, and a progeny is produced, and that progeny or offspring is called hybrid. Okay. So if you cut a hibiscus plant or a rose plant and you are planting it in your garden, and you have produced 10 rose plants or hibiscus plants, they cannot be called as hybrids, because they are produced not as a result of crossing two genetically different individuals. They are not produced as a result of cross-pollination. Instead, they are produced as a result of vegetative propagation. So, a hybrid is produced when two genetically different individuals are crossed to produce a progeny. That's why we can have a hybrid cow. The best common example in our locality is Swiss brown. Swiss brown. It's a combination of two separate, genetically different individuals, cow and a bull. Or we can have a G into D coconut in our farm, in our field. A tall variety and a dwarf variety being close together to produce a new one called G into D, high yielding disease resistant varieties. Okay, so hybrid is organism produced crossing two genetically different individuals. And depending on the number of characters possessed by the parents, they are monohybrid, dihybrid, and polyhybrid. So, in an experiment, breeding experiment, we are looking at only a single character. For example, color of the flower. For the time being, we are not at all concerned with any other characters. We are only looking at the color of the flower. Such a cross is called monohybrid cross. And such a trait is called monohybrid character. And we are considering two characters at a time. And if we are considering two characters at a time, then it is called a dihybrid cross. Two characters at a time, then it will be dihybrid cross. Example, height of the plant, whether it is a tall or dwarf, also the color of the flower, whether it is red or white. So we are considering two characters at the very same time, height and color of the flower. Am I clear? Yes. And third type is polyhybrid. Many characters are considered together. More than two characters. Trihybrid. And more, several characters are considered at the very same time. One caution is that as the number of characters increase, it will be difficult to interpret and collect the data. Okay? The data will be huge data. So, this is the different concepts which we are to be familiar with this course. Then what is genotype? Genotype and phenotype. Genotype is the genetic constitution of an individual. Okay? The genetic makeup, the genetic makeup of an individual, irrespective of whether the genes are expressed or not. We know it can be a recessive allele or a dominant allele. Only the dominant allele will be expressed normally, and whether it is expressed or suppressed, the genetic constitution of the individual is its genotype. Okay, it is genotype, and genotype cannot be modified by environment. Okay, so genotype cannot be modified normally by the environmental factors. And next term is phenotype. Phenotype is the expression of the genotype. That means it is the morphological, visible, measurable expression of the genetic makeup of the organism. So, if there is a capital T present in the genotype, its phenotype will be tall. It is expressed as tallness. That's a tall plant. And if a dwarf plant is obtained, if the phenotype is dwarf, then we can definitely say its genotype is small t, the recessive homozygous condition. Okay? So the phenotype can be modified by the environment. Okay? If the environmental conditions are highly favorable for the progeny, its phenotypic expressions may be modified. But it does not modify its genotype, the genetic makeup of the organism. Okay. So the important factor is the genotype. Sometimes some students in the schools especially, so they are very poor, just like our mentioned, maybe coming from very poor family, there is no facilities available. There will be very poor situation, so that they can send only these children to the public education system. The parents are not well versed to afford the high fees in the English medium or CBSE schools. But once these children are put in a very good condition where they are getting the chance of acquiring a knowledge, they will be excelling all the other fellows which are so-called the broiler grown children of CBSE or English medium schools. So the environment has changed, but his inborn genotype, he has the inborn capacity, innate ability to understand, study and excel. It was there, so he started getting in the forefront. Or certain people who are residing in very hillside, there won't be any chance of going for any practice in the sports field. Once they are put in regular practice with the good facilities and scientific training, they will come up with better and fine excellent results. That means they had the genes or genotype present. It was not conducive for them to excel. When they were put in the exact or good, apt environmental condition, they produced better results. That means their phenotypic expression was excellent. If a plant, coconut tree, which was starving in the soil of very, very poor nutritional conditions, but it has the genotype of high yield, but there is no possibility of getting no sufficient nutrients. And if the environmental conditions are changed by putting sufficient quantity of all nutrients, definitely the plant will start producing its full vital capacity. And on the contrary, if the plant is not having any ability to produce high yield, because its genotype is such that only few seeds are produced, even if provide all the suitable conducive atmosphere, water, nutrients, better aeration, there is no shade, everything, irrespective of all these environmental conditions, the plant cannot yield high yield because its genotype is not good to produce sufficient quantity of fruits. So genotype and phenotype, they are very important. The genotype is genetic constituent of the individual and it cannot be modified by the environment. Whereas phenotypic expression of the gene can be modified by the environment. Okay, so these concepts, alleles, allelum of gene locus, then factors, dominance, recessiveness, hybrid, genotype and phenotype. These are the key concepts. Monohybrid, dihybrid, polyhybrids. These are the key concepts with which we will be progressing in our next modules. Thank you.